Okay, thank you for coming back on time. So we will start this afternoon session in a timely fashion. And we're incredibly excited to have Marina Kuimova that's going to deliver a keynote lecture this afternoon. So please give me a couple of seconds to introduce her. Marina did her undergraduate studies in Russia and then went to the University of Nottingham in the UK, where she did a PhD with Michael George, studying the electron transfer in DNA in metal complexes. She then moved to Imperial College, where she was working with David Phillips, also doing transient absorption spectroscopy at the time. Imperial College obviously realized they had someone that was really special because they kept her all after that. And that's where she continued working for the rest of her career, and she's now a full professor there. I asked Grok to come up with 2 or 3 sentences to describe Marina's work to us, and asked him to do it for a 12-year-old student. And so here's what Grok has to say about Marina's work. So he says that Professor Marina Kuimova is a smart scientist who studies tiny things inside living cells, like how thick and sticky the goo is that surrounds our body's building blocks, kind of like checking if honey or water flows better in different spots. She uses special glowing tools and cameras to take quick pictures of these hidden spots without hurting the cells, helping doctors figure out why the sicknesses like cancer happen and how new medicines can swim through to fix them. Her cool discoveries also help us learn about big problems like how pollution in the air changes the weather. So I think with that, we know all about what type of work she'll be presenting to us, and please join me in welcoming Marina for her speech.
Thank you very much, Dario, for this very kind introduction. I should ask Google or ChatGPT to write my proposals for me. I think I'll get much better success rates. Well, it's an absolute pleasure to be here. And I was really, really impressed by the breadth and the depth of the activities as you have as part of the paper. You'll see from my presentation that my work aligns, I think, mostly with the themes of health, but also characterization of photochemistry and material sciences. I'm hoping that there will be some slides, otherwise I'll have to do chalk and talk. But let me continue talking and tell you that we are mostly interested in using fairly sophisticated microscopy and spectroscopy techniques to learn about the biological processes within cells. So we use fluorescence lifetime as the main readout of the behavior of our molecules. And we try to apply it to a large range of problems, mostly to do with diffusion within cells, but actually covering a large range of different problems. So my talk, I hope, will be a little bit like British weather. You know, every few minutes it will change, right? So if you're listening to what I'm saying and you don't really interested or you don't like it, just wait for 10 minutes and hopefully, you know, the next topic is going to be better. But I do need my slides, I think. The slides here, but they do not project. Oh, fantastic. Great. So, as I said, we are extremely interested in using microscopy and spectroscopy to look at the diffusion control processes within cells. So, for most of you in the audience, that diffusion and viscosity is really, you know, cells are extremely heterogeneous environments. There are lots of different organelles. Most of these organelles are then surrounded by lipid bilayers, a very hydrophobic border kind of control structures that then separate the organelles from essentially aqueous phase of the cellular cytoplasm. So in this complex system, perhaps it's not surprising that viscosity plays a large role in processes such as bimolecular reactions and signaling and diffusion. There's also really good body of evidence in the literature that abnormal viscosity is linked to disease and malfunction, both on the single cell and the organism level. So to us, this meant that trying to learn about hydrodynamic properties of cellular environments is really important to understand the role of viscosity in disease and malfunction. But actually, looking at viscosity on a single cell and organism level is really tricky, because the cell organelles are tiny, and traditional mechanical ways for measuring viscosity will be very destructive, and also they'll require large amounts of material. So, of course, there are lots of techniques that are spectroscopic that have been developed, shown here, that use the length scales of individual organelles to try to probe diffusion control processes within cells. So you'll recognize most of these methods, perhaps, monitoring diffusion controlled photochemical reactions that are viscosity dependent, or FRAP or FCS, so recovery after photobleaching, or correlation spectroscopy. But all of these methods, when we started looking at them, we realized that they have two major limitations. First limitation is that these methods mostly measure things in single points instead of providing us the full image, the full picture of what happens within cells. And second limitation we identified is that most of these methods, they require the measurements to be acquired over time, several points, and then processed to produce the viscosity value. And that means that you cannot measure viscosity in real time, just as the process of biological importance occurs. So what I hope to show you today, that our method, that is based on fluorescence detection from molecular rotors, overcomes both of these limitations. And we can get good spatially resolved pictures, and we can do it fairly fast. So if we want to, we can monitor dynamic biological processes that occur and cause changes of viscosity. Okay, so what are molecular rotors? They are small synthetic fluorophores in which there's a large degree of conformational change that occurs in their excited states. And this is extremely important because this conformational change, such as internal rotation, leads to a depopulation of the bright excited state and brings it to the dark, non-emissive state. So, if our rotors are in a non-viscous environment, shown here by water, they rotate very, very fast. The dark state is being accessed very efficiently, and as a result, you see very small amounts of fluorescence. However, if we place our rotor in a confined or viscous environment, the rotation doesn't happen, and then the dark state is not efficiently populated, and you see a very large increase in fluorescence as a result. So, listed here, there are several groups of molecular rotors that we investigated and discovered in the course of the last 15 or so years. But before I move further with describing how we use rotors, I really need to discuss the fact that fluorescence intensity is not a concentration-independent parameter. So you can put your rotor in a heterogeneous system, and then the intensity of your signal that you record does not just depend on viscosity, it also depends on how much of the rotor you have in a particular organelle within your system. And this is not good enough, because in something like cell, we have no good way of determining the concentration of our fluorophore. So what we need instead is some kind of ratiometric approach that takes this concentration into account. And we suggested just such an approach based on the fluorescence lifetime. So fluorescence lifetime is a characteristic of how fast the excited state decays back to the ground state. And this is concentration independent in the absence of quenching or self-aggregation. And just like fluorescence intensity, fluorescence lifetime increases in a very steep upward curve as we increase the viscosity of the environment. So we first discovered it for this particular molecular rotor that is based on the Bode-P structure. And our Bode-P is quite special because it has a phenyl group attached to the fluorescent core of the Bode-P, and the phenyl can rotate. And as it rotates, the dark non-radiative state is being populated. And so in a non-viscous environment, the intensity and fluorescence lifetime are very short. However, if you increase the viscosity, the lifetime and fluorescence intensity go up. So you can build a calibration plot, in a way, by measuring fluorescence lifetime in solutions of known viscosity, such as methanol glycerol or toluene castor oil or lipids of different composition of different temperature. So what is plotted here is the lifetime as a function of viscosity, and I plotted it in a so-called Förster-Hoffman plot, where it's a doubly logarithmic coordinates. And you can see that this plot for our BODP rotor is linear dependence. So it means it corresponds to a power law in a very large range of viscosities, so from 10 to several thousand, in fact, centipoise. So we think that our rotor will be suitable for measuring viscosity in biological environment, because biology, we hope, operates somewhere in between 10 and 1,000. Centipoise. But of course, you can modify the rotor structure or invent different types of molecular rotors that measure either lower or higher viscosity. If you, for example, had an idea to apply to material sciences where viscosities are in a different range. Okay, so one really important thing is that fluorescence lifetime is something that can actually be imaged in a very precise, spatially resolved manner using a fluorescence microscopy technique called FLIM, fluorescence lifetime imaging. So in FLIM, we essentially use the pulsed laser excitation from a Ti-sapphire or a diode laser, depending on what type of microscopy we're doing. And then we use single photon counting to detect time-resolved fluorescent decays in every single point of our image. So it's a bit slower than confocal or wide field imaging, because we have to build a fluorescence time-resolved histogram in every single point of the image. But then as a result, you can fit the data using multi-exponential or mono-exponential decay, extract the lifetime, and of course the lifetime unequivocally corresponds to a certain viscosity. So this way you can create maps of viscosity in cells, in tissues, in heterogeneous materials, in aggregating proteins or polymerizing systems, you name it. Okay, so what do we do in my group? We look at the structures of molecular rotors, we create new structures, we collaborate with others that can provide us new molecular rotors. And then we characterize them and try to apply them to a large variety of different problems. And today I'm going to try to concentrate on our efforts in probing viscosity and conformation in biological type of systems. Okay. So our first goal when we sort of invented molecular rotors was trying to look at viscosities of those lipid bilayers. So the plasma membranes and internal organelle membranes that are surrounding organelles in cells and providing a really important permeability barrier and defining mechanical properties of cells and tissues. So for this, our BODP rotor is particularly perfect because it basically has the right lipid-like hydrophobic structure that it seamlessly incorporates in lipid bilayers or monolayers. And then we can record fluorescence images, and I'll emphasize again, instead of just looking at intensity, in every single point of this image, we record a time-resolved fluorescence curve, and then by fitting this curve, we can assign the lifetime. And when you see in the image the sort of artificial color scale that shows blue lifetime, it means you're dealing with low viscosities, and you can assign the actual number to it. And if you see red areas, then it means that you're dealing with high lifetimes and really high viscosities. Okay, so it's a quantitative method. Okay, so then we took our molecular rotors and said, well, great, wouldn't that rotor incorporate into the plasma membrane of the cell? And then we can measure the viscosity of the plasma membrane as it depends on different environmental conditions, including treatments such as cancer therapy. But unfortunately, our first design of rotor decided to behave completely different, because cells are sometimes a bit like little children. So these cells decided to take up our rotor and internalize it. And instead of staining of the plasma membrane, we see a staining of individual hydrophobic organelles, which are lipid droplets within the cell. But still, it was really useful. It was the first result that we obtained with rotors. We recorded time-resolved decay of rotor that gave us a lifetime of about 1.6 nanoseconds, which, according to our calibration graph, corresponds to a very high viscosity of about 140 centipoise. So we know for sure that our rotor is not in the aqueous phase of the cell cytoplasm, which is about 1 centipoise. But instead, our rotor is in those lipid-like bilayer domains within the cell, which have really high viscosity and low diffusion. And we can corroborate it with alternative measurements, such as diffusion-controlled reactions or fluorescence recovery after photobleaching, or FCS. But now back to the drawing board, right? We still want our rotor that stains just the plasma membrane of the cell. So to achieve this, we can modify the structure by keeping the functional part of our fluorophore intact, and instead modifying the phenyl group with different substituents, such as cholesterol, or this doubly positive charge through coordinized amino groups that essentially acts as a stopper, right? So the charge prevents the probe from internalization inside cells, and then we hope that this will allow us to stain the plasma membrane. I should say also that for all the three rotors, we know from molecular modeling that even though the structure is a little bit different, and that determines their different localization within the lipid bilayer, their active rotating group is still in the same place, and so in model lipid membranes, they still measure exactly the same viscosity, which is shown here as a function of temperature. And importantly, if we mix our lipids in the right proportion of DOPC, DPPC, and cholesterol, so these are the three model lipids that probably make most of the living organism membranes, then we can have phase separation. So when our cells separate, our membrane separates into the liquid ordered and liquid disordered domains. And for each of these domains with these rotors, we can really determine the quantitative values of viscosities in those phases and look at their effect of composition or temperature on these values. Okay, now back to the cells. Of course, I wouldn't be telling you the story if one of these rotors didn't actually work. So the rotor which has the stopper groups, so the double charge at the end of the alkyl chain, actually doesn't penetrate, doesn't internalize within the growing cells, and it allows us to stain the plasma membrane of these cells very, very efficiently and study some effects of chemotherapy, for example, which I'll mention shortly, and other factors that can affect the health of our cell. Another more or less unexpected positive side effect was that because of the two positive charges, our rotor is now a lot more hydrophilic, and that's actually really positive for in vivo work with these rotors, because the original rotor that doesn't have positive charges completely accumulates in kidneys and liver of live animals, and we can't study anything to do with cancer in live animals using this molecule. However, this more soluble derivative now accumulates really well in live animals in injected cancer cells, so we can actually look at something really useful for example, for animal models for cancer with this molecule. Okay, so here we used this slight tweaking of the structure to introduce cholesterol or double positive charge to change the localization of our rotor in the cell, and this is really successful. But the question is, can we do even better? Can we pinpoint the localization of our rotor even more precisely? And it turns out we can do. So here it's done using a halo tag approach. So we can transfect our cells with transiently with the DNA that encodes for a halo protein that could be expressed in a particular organelle within the cell. We can design which organelle it could be. And then we can modify our rotor with something called a halo tag ligand, which really quickly and irreversibly covalently binds to the cavity of the halo protein. And so simply by incubating the transfected cells with our halo tag rotor, we can achieve really nice organelle staining using molecular rotor, achieving different organelles stained and viscosity monitored using essentially the same probe, just different target halo protein. Okay, remember when I started my talk, I said that we are looking to address two major limitations in viscosity imaging in cell. And one is that we are not happy with just measuring a single point within the cell, and I hope it demonstrates to you that we can address this by targeting the rotors and by doing FLIM images of our molecule. But the second limitation was that we would like to be able to do dynamic measurements of viscosity in a cell just as something changes, that is biologically and medically relevant. So it's important that when BODP or any other rotor that we use is in a viscous environment, the fluorescence intensity is fairly high, and so we can accumulate our FLIM images within 1 or 2 minutes most of the time. And that means that we can image changing viscosity within the cell if the process that we are interested in, of course, is slower than the rate of our FLIM image acquisition. So we are limited to this, 1 to 2 minutes, or maybe half a minute in best cases. But we can monitor dynamic processes within the cell just as they occur and the viscosity is changing. Okay, so just to give you some examples of this. So this is a monolayer of HeLa cells that were incubated with cisplatin to basically kill them using chemotherapy. And we can simultaneously monitor the viscosity within the plasma membrane of these cells, and at the same time we can extract lipids from the plasma membranes of these cells and analyze them using mass spec. And we see in both cases with fluorescence and with mass spec, we can see really drastic, statistically significant changes. So for example, upon action of cisplatin, initially within maybe 10 minutes to 1 hour, viscosity drops, and we see also an increase in unsaturated lipids extracted from the cells. However, if we continue growing the cells upon action of cisplatin, we see the opposite change. The viscosity of the cellular membrane increases dramatically. And we can reproduce these results in spheroids and even in vivo model in mice, which is done in collaboration with my colleague Marina Kuimova in Nizhny Novgorod in Russia. If we take a different chemotherapy agent, so here we look at Taxol, we actually don't see any increase in viscosity at all, and we see a decrease in viscosity. And we can again analyze the cells using mass spectrometry, and we now are fairly sure that this decrease is to do with the Taxol action as an agent that breaks the links between the membranes and cytoskeleton. And we're pretty sure it's not the lipid composition, but this breaking of the cytoskeleton to membrane linking proteins that causes the increased fluidity, so decreased viscosity of our membranes upon treatment. Okay, so another example of dynamic monitoring of viscosity. In this case, we decided to look at something unusual, namely the release of carbon monoxide. So we all know CO is a toxic molecule that attaches to hemoglobin, as we heard from Majed earlier today. But also CO is a very important signaling molecule within the cells, and it's being released during inflammatory response, for example. Well, for this study, we teamed up with my colleague at Imperial, James Wilton-Neely, who has a really keen interest in monitoring CO in cells. And he does that by using transition metal complexes, for example, osmium or ruthenium. And the principle of his probes is that the initial complex is dark. However, when the CO is attaching to the ruthenium sensor, due to the trans effect, the fluorescence switches on really, really intensely. So what we've done together with James is we actually attached one of our rotors, the BODP, as the ligand of the complex. And we realized that not only the fluorescence switches on when the CO is being attached to the complex, but also the fluorescence lifetime of our BODP still reports really well in the viscosity of its surroundings. And so here, there are two series of cells treated to induce the formation of CO in them using two different ways. One is a CORM, a chemical based on transition metal complex that releases CO within the cells without starting any metabolic processes or any oxidation reactions. So you just see a large increase in CO within cells. And in this case, we see a large increase in fluorescence of our complex, but no change in lifetime, because we don't expect viscosity to increase or change or be altered in any way. However, if we incubate our cells with hemin, which is a molecule that acts on the iron hydrogenase enzyme that starts the oxidative reactions within the cells and produces carbon monoxide through these reactions, we actually see even larger increase in fluorescence, which means that even more CO is being produced intracellularly. But this time, we see a large decrease in viscosity at the same time. And this to us means that we are probably producing reactive oxygen species within the cell that are consistent with breaking the double bonds within the lipids and probably affecting the protein surrounding the rotor as well, which leads to lower viscosity as a result. Okay, so this is a really crazy experiment, but I've been contacted one day in my office by a space scientist from European Space Agency who read my papers, and he said, let's measure viscosity in space. So we never really quite managed to get the space work funded, but instead a couple of my students applied for funding from the program called Spin Your Thesis, and had access to a huge centrifuge that is localized in Netherlands at the European Space Agency. And essentially the centrifuge is so huge, and it can go up to a huge gravity conditions of about 15 to 20 G, but it's so large that you can fit the whole microscope in one of the gondolas of the centrifuge. So we applied for the Spin Your Thesis time, we got it funded, and we traveled with our cells and molecular rotors to this ESA facility, and we incubated our cells on the microscope stage and then took them up to 15 G, as you can see here. So in green, you see the ramp of gravity, and in black, you see the fluorescence intensity change as the gravity is being taken up and then down again. And we've had really nice reproducible results for several cell types that show that the viscosity within cells follows the ramp of hypergravity. So viscosity increases dramatically as you spin the cells, and then it goes to a large degree down. However, if you just extract lipid vesicles from the cells, from the plasma membrane-derived vesicles, you don't see these changes at all. So it's really the effect of hypergravity, like pressure, on cytoskeleton of the cells. So it's really, really interesting, and would be nice to be continued in space, but it's so hard to get it funded, so we did not yet succeed. Right, so I know many of you in this audience are really interested in photodynamic therapy, and so are we. And so I decided to show some slides that show that molecular rotors are actually good reporters in looking at what reactive oxygen species such as syngas oxygen or radicals do to viscosity within cells. So I don't really need to describe in detail for this audience, but just in case if there are some material scientists that don't know this, PDT is a clinical way of treating neoplastic diseases such as cancer with fast-growing tissue. And they include administration of a drug called photosensitizer to a patient. But then due to various processes, for example, lymphatic drainage or targeting, the photosensitizer specifically localizes just in cancerous tissue. But it's not doing anything unless you shine light on it, red light in this case, and then the photosensitizer is being activated by this red light and starts producing reactive oxygen species, such as syngas oxygen, type 2 photochemistry, or radicals, type 1 photochemistry. And these species are very oxidizing, and they destroy cancerous cells and tissues through the redox reactions that they start, and then that results in removal of the fast-growing tissue in healthy patients. Okay, so for this particular work, I'd like to talk about a molecular rotor that is also a photosensitizer. So this dye here is constructed as two conjugated porphyrin dimers linked through this conjugated butadiene bridge. And the two porphyrin units are in conjugation, however they can rotate. And it turns out that the rotation between the two units is viscosity dependent. But what's important to know is the conjugated form of the dimer, when the two units communicate, is the lower energy emission that emits at about 800 nanometers. However, if the two units are twisted, the communication and the conjugation between the two units is broken, and the emission from that state is at high energy, a twisted kind of state, at about 700 nanometers. So if you now imagine that you excite the twisted state, which is high in energy, it has two options. Either it emits light at 700 nanometers, or it rotates to a more stable planar conformer, and then the planar conformer can emit at 800 nanometers, right? So what we discovered is that you can calibrate the fluorescence responses of the porphyrin dimers based on viscosity. And in low viscosity environments, all the twisted excited state rotates into the planar, and you see predominantly the emission at 800 nanometers. However, at increasing viscosity, you see more and more progressively emission from the twisted state, and you can build a calibration plot of the dimer by dividing the intensity at 700 to the intensity at 780. And as you can see, again, it shows a really nice calibration curve in this doubly logarithmic Förster-Hoffmann coordinates over several orders of magnitude. So what we're going to do now is we're going to get our dimer to produce syngas oxygen, because of course it's 2 porphyrin units linked by a bridge. And so it's very efficient at producing syngas oxygen. But at the same time, we're going to measure the ratio of its emission at 700 to 780 and record the viscosity just as it changes as our cell dies as a result of photodynamic therapy. And we saw a very large effect. So in a healthy, non-perturbed cell, the viscosity is around 80 centipoise. However, as you continuously irradiate it and the cell eventually dies, the viscosity rises sharply until it plateaus at about 350 centipoise when all the photoinduced processes in the cells are complete. So this was really cool. And over the years, we answered ourselves some questions. You know, is the porphyrin dimer unique? Can we think of other molecules that do this? And the answer is yes, but I'm not going to talk about this now. But what I wanted to talk about today is what is really the mechanism of this increase? Why does this happen? So for this, we decided to separate the function of the photosensitizer and the rotor. And we're going back to our good old BODP molecular rotor that is particularly good for measuring viscosity in lipid bilayers, right? And we're going to use a photosensitizer, which in this case I think was PPS, so a hydrophobic sensitizer that goes to the lipid bilayer nicely. And so we look at the large number of giant unilamellar vesicles that are large enough in size that you can see them using optical microscopy, and then we zoom in on one vesicle at the time and we irradiate it, and we see the changes in viscosity of that vesicle using BODP fluorescence lifetime. And so I hope you can see here very clearly that as you irradiate, the viscosity of this vesicle increases very, very substantially. And importantly, it doesn't happen if you add a quencher such as sodium azide. And it also doesn't happen if you make your lipids out of unsaturated, out of saturated lipid, which has, for example, methyl groups rather than double bonds. So has the same viscosity, but doesn't have anything that could be oxidized. On the other hand, if we now make our vesicles in the same way with BODP, but we use a different photosensitizer, so in this case methylene blue, that is known to produce a large amount of radicals as well as some of syngas oxygen. Then by zooming on a single vesicle, you actually see the opposite trend. You see a decrease in viscosity. And we know that this is because the radicals that are being produced are a lot more reactive to syngas oxygen. And instead of forming peroxides and polymers out of lipids, they actually cleave those double bonds, and that results in a decreased viscosity and a higher permeability of the lipid bilayer. We actually recently were able to produce those vesicles artificially. So we produced, with the help of Carlos Marcos, we managed to produce those vesicles made of completely oxidized, peroxidized or cleaved lipids, and perform molecular dynamic simulations to confirm these conclusions. So we know type 1 photochemistry causes a decrease in viscosity, whereas type 2 photochemistry causes an increase. So now you can rationalize our data for PDT. In PDT or in cisplatin treatment of cancer, we saw a type 1 peroxidized kind of photochemistry that increased the viscosity. However, in the presence of methylene blue or in the presence of heme that produced the Ross response within cells, we saw a decreased viscosity, which is most likely linked to the radical production. Okay, so remember my kind of bid to do this measurement more targeted. So we can also make our sensitizer more targeted. So in this case, we are going to choose the genetically encoded protein called killer red that could be encoded in any particular organelle within our cells. And we are going to then choose at will which organelles we're going to irradiate and which organelles are going to be flooded with reactive oxygen species. So this picture here is to show that this is work in 2011 that killer red was demonstrated to produce mostly radical species, so no syngas oxygen at all. And so by adding killer red to our cells, we most likely produce reactive radicals that are going to initiate type 1 photochemistry. Okay? So this is, again, the work in collaboration with Marina Kuimova, and they're very good at producing this encoded cells with killer red, in this case, that could be localized in 2 different organelles, either in plasma membrane here, or in the nucleus of our HeLa cells, right? And so in both cases, we're using BODP that is localized in the membrane. So our readout is in the membrane, but our initial oxidative attack using killer-red irradiation that produces reactive oxygen species is going to be different by organelle. And so here's the viscosity data, and what you can hopefully see from here that killer-red localized in the nucleus actually causes a decrease in plasma membrane viscosity over time, whereas the plasma membrane localized photosensitizer causes an increase of viscosity. And this has been confirmed in cultured monolayers and also in spheroids. So that's pretty interesting, that different localization of initial oxidative attack can really change the course of what's happening to the cells in terms of their permeabilization and diffusion control processes within them. Okay, now let me try to change gear a little bit and tell you about a different health-related process to do with protein aggregation. So we all know about amyloid-related diseases when sometimes benign and soluble proteins, sometimes for some reason, starts to misfold abnormally, and that promotes its oligomerization and finally aggregation that can result in really serious health consequences in amyloidogenic diseases such as Alzheimer's and Parkinson's. So it's really well known that the mechanism of this sort of amyloidogenic aggregation is really complex, and it differs from protein to protein. And that makes it very, very difficult to study, especially because what's considered the most toxic species, the prefibrillous small oligomers, are not easily detectable by any kind of standard technique. So the gold standard of detecting amyloid aggregation is by adding a fluorescent dye called thioflavin T, or THT for short, that binds really strongly to fibrils, to beta sheets of fibrils, and it makes it light up and it reports on aggregation this way. So we hypothesized that if we use our molecular rotors, that even if they don't specifically bind to oligomers and fibrils, they'll be confined by the increasing crowding that is created by those oligomers and fibrils eventually being created. And we'll hopefully see a large increase in fluorescence lifetime as the aggregation occurs. And that's exactly what happened. So in fact, even the THT is in itself a molecular rotor, and even its intensity only reports on the fibril formation. The lifetime, it seems, can report on more intermediate stages, early stages of aggregation. So I'm only showing one sort of data graph here, but this is the so-called phasor analysis of the time-resolved decay traces shown here that correspond to aggregation of lysozyme, the protein involved in the disease called ALS. Here. And so I'm not going to go in detail through this sort of data analysis, but what I want you to notice here is that this trajectory of phases is not linear, and that means that it's not simply interconversion between two species, A and B, so free monomer and aggregated fibrils. It definitely has some other stages here that we call 2 and 3 and 4. And so what we can then do is use alternative techniques, a bit like Majid's example with the elephant, in blind people. So we use different techniques to try to understand what's going on. And so we can use single molecule AFM and nanopore detection to identify that actually what happens here is a decrease in viscosity that is caused by fragmentation of our mature fibrils that are formed of lysozyme. The slide here actually shows very similar data, which is hot off the press, recently published, when we looked at aggregation of alpha-synuclein, which is a protein involved in Parkinson's disease. And here we use two molecular rotors, which are both commercially available molecule, THT and DISC, which is a derivative of cyanine-free dye. And so while intensity of these dyes pretty much only responds to fibrils formation, we figured out that the lifetime of both of these dyes reports at much earlier events. It rises a lot earlier. And if we look at the phasor plots of these aggregations, the red points here that are due to aggregation of alpha-synuclein wild type, actually these points that we see on phasor plot cannot be matched by any kind of control conditions of producing mature fibrils, shown here in black, or oligomers type B, which are fibrillar oligomers. And so that tells us that these are due to this really early prefibrillar aggregates, oligomers type A, that have not been easy to detect at all using any techniques. And so we are now actively pursuing this work, looking at variants of proteins that are pathological and involved in, say, familiar forms of Parkinson's. Okay. When we look at molecular rotors, we worry a lot about temperature. And this is because the way they work is, in order for fluorescence to deactivate via population of the dark state, the molecular rotor normally has to overcome an energy barrier before it ends up in the dark state, which then becomes deactivated. So it's quite conceivable that changes in temperature, as well as changes in viscosity, can affect the photophysics of our rotor. So how do we counter for this? Well, the way that we try to control for this is we do control experiments, for example, shown here with BODP. When we produce solutions of methanol glycerol of changing methanol glycerol content, so the higher glycerol, the higher the viscosity, and also change in temperature. So the higher the temperature, the lower the viscosity. But importantly for all these solutions, because they're bulk solvents, they're Newtonian liquids, we know the viscosity of them. And so we can measure the lifetime and produce this calibration curve of lifetime versus viscosity for all these different compositions in different temperatures. And I hope you agree with me that the overlap is actually pretty good. So from this we conclude that at least for BODP rotor, it doesn't really depend on the solution composition or solution temperature, and what it measures is exclusively viscosity, which is great because it allows us to use this rotor for temperature-independent measurements within the cell. So we can study the cell at 10 degrees or 37 degrees, and both times we'll get different viscosity values, but it's not because the temperature is different, it's because the viscosity is different. Okay, so then we thought to investigate a different structure of BODP. In this case, you see that it has extended conjugation. And the reason we've done it is because we thought we'll make a red-emitting rotor. But actually, what we actually got is the dyes where, by changing the substitution, we changed the height of the barrier between the dark and the bright state. And so for this extended conjugated BODPs, especially BODP3, the barrier is the highest. And as a result of this, our new BODP is now not a molecular rotor at all, but instead it completely depends on the temperature of its environment. So we wanted a red-shifted rotor, but we actually ended up with molecular thermometer, which is also quite useful, especially because due to the small size of the substitution, it's cell permeable. And so we can now measure temperature within the cell if we want to. The accuracy of this measurement is not terribly high, so it's probably not really good enough for detecting the temperature change of any biological process, such as cancer or amyloid aggregation in the organelles. But it gives us hope that with further modifications of the structure, we can potentially create more sensitive rotors thermometers that work by fluorescence lifetime. Now, going back to our conjugated porphyrin dimer, you will remember that it works ratiometrically. So we have two peaks at low wavelength and the high wavelength. Depending on the substitution, it could be 6 and 700 or 700 and 800. But so this one is 650 to 700, two peaks. But what I didn't tell you before is that this rotor is also a lifetime rotor. So if you measure the fluorescence decays in the twisted peak of the decay of the porphyrin dimer, then you'll be able to measure the viscosity using FLIM. So what we then set out to do is to check how the temperature affects the photophysics of our conjugated porphyrin dimer. And the results were very surprising. Well, first, not so surprising results. If we measure the fluorescence spectra in methanol glycerol solutions of different composition and different temperature, we actually have a perfect overlap of all the curves, even better than BODP. So there's no doubt in our mind that if we use ratiometric response of the fluorescence dimer, we get viscosity measurements without any effective temperature. However, the story was completely different when we looked on the fluorescence lifetime. You can see here, especially for the dimer 2 and 3, so the structures are shown here, and they just change in the degree of biocompatibility, so how well they work in cells. So for 2 and 3, you can clearly see that the curves recorded for different compositions and different temperatures do not overlap. So when my very talented student, Arimas Vishnauskas, did this experiments, he was actually really disappointed. He thought it's an artifact and something is wrong, until we realized that we can actually fit all this data using a type of Arrhenius dependence with the activation energy. And so then what we have is we have a single dimer that you can measure the fluorescence lifetime and fluorescence spectra of. The fluorescence spectra depend only on viscosity. So by measuring the spectra, you know the viscosity for sure. And then you measure the lifetime, and the lifetime is, in a very nice calibration framework, depends on viscosity and temperature. But you already know the viscosity from your spectral response, right? So you can now use all the data to figure out the temperature and viscosity simultaneously. And to the best of our knowledge, this is the only type of molecule that they can do it in one measurement. And this could be important because if you can imagine a viscous environment, such as organelles in cells, and various temperatures will really, really change the viscosity of that environment. So you can't just assume that fixed viscosity is maintained for our temperature variations. So this could be a handy tool for measuring both parameters simultaneously. I still have a bit more, but do I have time, or do I need to stop? Okay, that's perfect. Right, so far, I used my molecular rotors in such a way that they were freely rotating within an environment of interest. And then the lifetime of our rotors told us about the diffusion-like, viscosity-like environment within the cell. So in the last part of my talk, I'd like to talk about the case where we actually want our rotor to bind tightly to an intracellular molecule. And then the lifetime will depend on the conformation of the binding, and it tells us something interesting about the conformation of the biomolecule, namely DNA in this case. So most of us know that DNA is present in the double helix within the cell, which obviously is responsible for the storage of genetic information. But sometimes this helix has to unwind. And if the helix is rich in a particular nucleobase, specifically guanine, it can form in really strange and interesting secondary structures, such as, for example, G quadruplex. So G quadruplex is a tertiary composition of guanines that are linked through Hoogsteen rather than Watson-Crick bonding. And we are going to use our molecular rotors binding to this unusual DNA structure, quadruplex, and because it binds in a completely different conformation to the way it binds to a duplex, we'll see a different lifetime, and hopefully that will tell us about the presence of G quadruplex within the cell. So why is it interesting? Because actually there are many cellular processes in which double helix has to unwind within the cells. And the efficiency of these processes regulate disease. So for example, the lengthening and shortening of telomeres is implicated in longevity and cancer. Transcription and replication are all very important processes, and quadruplexes are known to form during all the cellular processes, and of course they are tightly regulated by many proteins that are associated with them, etc. But detecting quadruplexes themselves is really, really tricky. There are a couple of antibodies that are available, but they can only work in fixed cells because you have to permeabilize your cells in order to get the antibodies in. So we are going to suggest a molecular rotor that is a small molecule that can work in live cells. And so in this case, let me introduce you to trianguliniums, which are aptly named for their triangular structure. But our triangulinium molecular rotors are quite special because they have these morpholino arms hanging off the structure. And due to hydrogen bonding interactions with quadruplex, morpholino groups will be taken away from the core of the triangulinium. And that means that when our molecule is bound to quadruplex, the quenching from morpholino groups to the core is almost completely eliminated, and we end up with a very long fluorescence lifetime, which is a lot longer than that in the duplex, shown here in green. And both of them are much longer than the lifetime that you see when the molecule is free in solution, and the arms are flapping around and quenching the triangulinium core. Okay? So, of course, we can calibrate against many DNA and RNA topologies. The quadruplex is shown here in red, and they all have very long fluorescence lifetime. And, of course, we can then perform FLIM, fluorescence lifetime imaging, where red pixels indicate the presence of these quadruplexes in cells. So what can we do with this kind of technology? well, we can screen, for example, for drugs that are non-fluorescent in themselves, but are suspected to bind to G quadruplexes. For example, here, green banana is our molecular rotor, and it binds to quadruplexes and produces a long lifetime. However, if we incubate our cells with a drug, but potential drug, which is the yellow banana here, if the binding is tight enough, then the yellow banana will bind and kick out our daughter triangulinium probe from the cells, from the quadruplexes, and the lifetime will shorten. And that's exactly what we see here in this titration with PDS, pyridostatin, which is a well-known strong G4 binder. So we know it for PDS, but we might not know it for many other drugs. Whereas if we use duplex binder, DAPI, here, it's known not specifically to bind to quadruplexes, and so there's no change in lifetime in this assay. We can also test for the enzyme activity. So, for example, many enzymes such as helicases are known to unwind quadruplexes at telomeres. So if helicases are active, quadruplexes are being unraveled, and you end up with a relatively short lifetime. However, if you knock out helicases from your cells, then the enzymes obviously are not present, not active anymore. You'll see an increased amount of quadruplexes and an increased lifetime, and that's exactly what we see. And we tested several helicases, and we even saw the effect of knocking out 2 helicases at any one time, and then you see a double increase in lifetime as a result of this. So a good activity test, potentially, which is not available via other means. We also have a specific probe that binds to RNA in the cell, so we can test for RNA quadruplexes and their effects through the fluorescence lifetime. And I think this is it, actually. So I hope I persuaded you that molecular rotors are a really useful tool to look at a variety of biological problems using basically spectroscopy and imaging, and also smart probes that can be designed for a particular application in mind. If you want to know more, we recently reviewed this for molecular rotor viscosity measurements and for quadruplexes. Both are in Angivante right now. And of course, all the thanks go to my wonderful group of postdocs and students who work really hard on molecular rotors, and funders and collaborators. And I leave you with some pretty pictures, and thank you for your attention.
So, thank you very much, Marina, for a very wonderful talk. I see we have lots of hands showing up, and microphone over to Margaret.
Thanks, Marina. Very nice talk. I have a small question concerning these zinc dimers. The doublet that you see around 650, 700 nanometers, this is an excitonic splitting?
So I'm not the right person to ask, but so. Both the Q band and the SORE band of the porphyrin dimer is split, and that's because of the symmetry. And it has been established through experiments and calculations that if you stabilize the dimer using the bridging ligand in a planar conformation, there's no splitting, just a planarized form. However, so the 650 band, the twisted band, it characterizes all the different orientations of the dimer that are different at about 50 degrees and more. So at 50 degrees, the conjugation breaks, and then all these isoforms, not one form, obviously, but all of them are characterized by this higher twisted band. There's lots of theoretical and experimental work from the group of Harry Anderson and his collaborators, and Harry, of course, is the one who made all the dimers.
Thank you.
Yes. Dave Thomas from Uppsala University. Thank you, very interesting talk. So as you heard, this is not my field of research, but I was thinking about this complex environment where, in membranes, for example, where I could expect that the viscosity or diffusion is anisotropic. So if you measure rotation in one direction, will you get the same number for viscosity as if it would rotate in a different direction compared to the plane. Is this something, a problem or something you can utilize or something?
It's a great question. So we have to be really careful how we define viscosity. You know, when we measure viscosity in a homogeneous system and solution, then it's a true viscosity and it correlates one-to-one to our methanol glycerol calibration. However, in the membrane, of course, it all depends on the position of the rotor on the z-axis. So if you are really close to the head groups of the lipid bilayer, then viscosity is really low, actually, and probably the function of the rotor is going to be affected a little bit by interaction with other molecules. So, for example, the proteins that are membrane embedded or water that penetrates the bilayer. However, for all our molecular rotors, we know that they're deep enough within the tail region of the lipid bilayer. So essentially, they behave as if they're in a tiny test tube that is made of lipid tails. And so they literally sense the diffusion. And we can correlate it with the diffusion measurements using FRAP or molecular dynamic simulations at many temperatures, and we know it works. But change the rotor structure, and you have to do the whole characterization again, because you might not know.
Thank you.
Thank you very much for the great overview of these different rotors. But the triangulum, this one was not a rotor. Right? What is the mechanism for its fluorescence quenching?
So it's photoelectron transfer dye. But the reason I call it rotor is first because it makes a good story. And secondly, it's because it's viscosity dependent. So you can correlate the lifetime to viscosity, and you see a really nice correlation. The thing is that it doesn't get a chance to be a rotor, because most of the time, when it's in the presence of the DNA or RNA, nucleic acid, it just binds to either duplex or quadruplex, and then it's not rotating anymore. And it's the position of the morpholino groups that are PET quenching the triangulinium core, right?
So the position of the external groups allow proton transfer or not?
They do.
Okay.
So if it's in access, if it's in solution, then the groups are kind of fluctuating due to Brownian motion, and the quenching is possible. However, if you are bound to duplex or quadruplex, the groups are taken completely away from the core, and then this PET process clearly happens through space, because then there's no quenching at all. You can also remove this quenching by protonating morpholino groups. So we had to be quite careful that that's not what happens, because you can also protonate, and then the lifetime goes up even higher, because PET is not energetically favorable then. But importantly, and we are trying to work this out, we haven't quite done it yet, if you change the structure of the triangulinium a little bit by extending the conjugation, so it's the sort of stuff that Bo Larsen does, it doesn't go to the nucleus of the cell anymore, and instead it localizes in cytosol and mitochondria, and it doesn't bind to nucleic acid, and it seems that then the lifetime is determined by viscosity. So we kind of are trying to then make them viscosity probes as well.
Tonio?
Yeah, thanks a lot for this inspiring talk. I have just two short questions. The first one is about the G4, and my question was, can you also imagine to have a probe which could be dependent or sense the difference in topology of the quadruplex between parallel, anti-parallel, or hybrid, for instance? And my second question about the rotors, the first part of the rotors in the membrane. I mean, you are inducing this rotational movement, so are you also, in a certain sense, inducing some hyperthermic effect in your membranes?
Okay, so the first question about quadruplexes, it's a really good question, and we're thinking a little bit about that. But so far, we see the largest difference between free in solution, bound to duplex, and bound to quadruplex. And all the quadruplexes, whether it's DNA or RNA, actually are all pretty much on the same level. We had a candidate rotor, which is a helicene dye with morpholino group. And then helicene is obviously chiral. And we found that the binding of helicene is really different for different types of chiral quadruplexes, especially if they have an overhanging group on top that really prevents the binding. But unfortunately, then helicene didn't work for lifetime. So the binding is selective to different type of quadruplexes, but the lifetime isn't. So we don't have a selective rotor for a particular type of quadruplex. All of them seem to respond particularly well with lifetime, so really high lifetime to c-MYC, which is an oncogene, quadruplex, and is really commonly found in, U2OS cells or bone cancer cells. I hope I answered this part of the question. So the hypothermia question for the membranes, so we excite our rotors either using 1 or 2 photon excitation, and this produces mostly the excited state that deactivates really fast within, several nanoseconds. So we don't actually see any temperature increase associated with that, but I'm sure, there's some schemes, but you can induce the thermal effect by, for example, attaching rotors to the surface of the plasmonic nanoparticles, etc. But we haven't, it's an interesting idea because you can kill cancer, obviously, using temperature generation, but we don't see it with rotors as far as I know.
Thank you very much for this very, very nice talk. I just have a small question about the polarity. Is there any influence of the polarity onto your rotors?
Yeah, so really, really good question, and it's one of our main controls. So for BODP rotor specifically, which localizes in the membrane, we actually spent a lot of time, because it was the first rotor, characterizing it in different solutions. So if you do the calibration in methylene glycerol or in toluene castor oil, at higher viscosities from like 50 centipoise onwards, you see no difference whatsoever. So it looks like the predominant factor is viscosity rather than anything else. However, at low viscosities, I think the barrier becomes small enough that it's the position of the dark state that also affects the decay. And it looks like the position of the dark state is affected by the polarity. So if you measure methanol versus toluene, which are both similar polarity, similar viscosity, but very different polarity, you see a huge difference. So 600 picoseconds to 100 picoseconds.
Okay, I think we're running out of time for questions. I'd like to please join me, ask you to join me in thanking Marina for a very wonderful talk.